go ahead. Thank you.
Sure. Great. Thanks, Dennis. It's a great pleasure to sort of talk about some of the work that we have been doing. So in terms of screen, right, okay. Right, so I'm going to talk briefly about genome-wide meta-analysis of cognitive empathy. And we define cognitive empathy fairly broadly, and I'll sort of explain why I say that a bit later. So I work with someone called Simon Baron-Cohen at the Autism Research Center in Cambridge. So let me start off by talking briefly about autism and explain what autism is. So clinically, according to ICD-10, which is WHO's international classification of diseases, autism is characterized by a triad of impairments. So autistic individuals usually have difficulties in social interaction. They also have unusually restrictive and repetitive behaviors and stereotype interests. And the third area of difficulty is in verbal communication. Now, the verbal communication aspect is no longer there in the DSM-5, and it's there only in ICD-10. So that's the clinical criteria for defining and diagnosing individuals with autism. But in addition to that, many individuals have a whole host of difficulties in different domains. I think it was Lorna Wing who said, if you see one person with autism, then you've only seen one person with autism. And I think this speaks to the underlying immense phenotypic heterogeneity in this condition. Some of the more commonly observed other characteristics in individuals with autism are things like lack of eye contact. This has been documented as early as 18 months. Amy Klin did a couple of fantastic experiments to show that infants who later went on to develop autism had atypical attention to social stimuli. Many of the individuals with autism also have oversensitivity or undersensitivity to auditory, tactile, and visual stimuli. Many individuals have difficulty dealing with changes in routine. So these are just, some of the difficulties that many of these individuals have. We also know that autism is largely genetic. In the 1970s, Michael Rutter conducted a very famous twin studies experiment, which identified concordance in monozygotic and dizygotic twins. There were some early clues that autism was genetic. If you looked at the broad autism phenotype in family members of individuals with autism, I think as early as the 1960s, you could see that family members of individuals who were diagnosed with autism had difficulties in verbal communication. They either had delayed development of verbal communication or they had some sort of associated phenotypes. So in the 1970s, you had the first twin study, but subsequently you had many, many more twin studies. And I think it was last year or the year before when there was this large meta-analysis of twin studies, which showed that approximately twin heritability was approximately 75 to 80%. So this was a nice review that came out, written by Thomas Bougeron, which sort of showed the development of twin studies and quantification of heritability estimates over time. So I think it's quite clear in literature that there is a considerable proportion of the variance in the condition that is attributable to genetics. But then if we were to parse this out a bit more, we want to ask what proportion of the variance is accounted by common genetic variants. So things like common single nucleotide polymorphisms, which have a frequency of greater than 1% in the general population. There were quite a few studies that came out. The one to the left, the two sort of plots over there, are papers from Clay et al. In 2014. What they have done is they have used GCTA to estimate SNP-based heritabilities in quite a few different cohorts. The first cohort that's shown, that is panel A, is the Simon Simplex Consortium, which essentially is a consortium comprising of one affected individual and two unaffected parents. You can see that the heritability is approximately 40%. The second one is a slightly larger cohort called the AGRI, which is the Autism Genetic Resource Exchange, which comprises individuals from both simplex and not so simplex autism families. And over there, again, you sort of see a similar heritability estimate of approximately 50%, I think. There was a very interesting paper that came out in 2014 in Nature Genetics, Cochler et al., which used a host of different methods to try and parse the heritability explained by different types of genetic variants. And what they show was that the bulk of the genetic variation lies in common inherited variants. So essentially things like SNPs. A small proportion of the variants can be attributed to rare de novo variation. So these individually explain a large proportion of individual liability, but additively, they don't explain a large proportion of the variants. There's also a large chunk which is unaccounted for. So this can be a whole host of factors, including environmental factors or things like hormones. We don't really know what exactly lies in this 41%. So that is autism. But in NIMH, I think at the turn of this decade, came up with this very nice idea called the research domain criteria. The research domain criteria was essentially how do we look at all these conditions and interpret them not purely on a case control manner, not purely based on what DSM-5 or ICD-10 defines these conditions to be, but in terms of underlying endophenotypes or biotypes, if you will. And this idea was to sort of look at these underlying traits and to see how they sort of explain differences within a condition and also differences between different conditions. And they were able to classify different types of domains, and one such functional domain that they identified was known as systems for social processes and cognitive systems. And under that, they mentioned the reading the mind in the eyes test. So before I talk about reading the mind in the eyes test, I thought I'd take a brief second to talk about empathy. Empathy is one of those really abstract concepts, which I know many people have been struggling to define. Broadly speaking, empathy can be referred to the ability to understand, share, and respond appropriately to thoughts, emotions, and feelings. You can imagine that if you are someone who isn't able to do this, both if you're someone who sort of shares more than what you should, or who isn't able to share as much as you are expected to, you can have difficulties in social interactions. And this can later contribute to reductions in subjective well-being. Empathy can be broadly divided into two aspects. So one aspect is cognitive empathy. So cognitive empathy is the first part of that definition, which is the ability to understand and identify emotions. And the second aspect is affective empathy, which is the ability to respond appropriately to someone's thoughts, emotions, or feelings. And they're both right. Can you hear me?
Yes, we can hear you again.
Okay, perfect. Sorry, I think that it got disconnected in between. Right, so let me go back. So Simon Baron-Cohen developed a test of cognitive empathy called the Reading the Mind in the Eyes test. The Reading the Mind in the Eyes test is a short test, an online questionnaire, where you are presented with photographs of people's eye regions. So you can see that over here. And you're also provided words describing the emotions that you're trying to portray. So things like playful, comforting, irritated, bored, etc. And the scores on this, the aim of this test is to describe the correct emotion that people are portraying purely by looking at people's photographs of people's eye region. So in case you're wondering, the first photograph over there responds to being playful, I think. The second one is terrified. The third one is desire. So different people score differently on this test. And if you were to sort of plot the scores of people, you see that it is generally a Gaussian normal distribution. There are two other things that the first paper described. One is that individuals with autism tend to score, on average, slightly lower, but significantly lower than individuals without autism. And the other thing that the initial paper described was that women tended to score higher, significantly higher than men on the ICE test. And these results are fairly robust. They have been replicated and validated in different studies using different versions of the ICE test and in different languages as well. So we were interested in looking at the genetic architecture of the ICE test. So what we did was we collaborated with 23andMe, who were very generous and used the full version of the ICE test on close to 90,000 participants of primarily European ancestry. We were also very fortunate to get a second smaller data set from the Queensland Institute of Medical Research. They used a 14 version of the ICE test on 15,500 individuals, and all of them of European ancestry. So using both the data sets, we were able to do a genome-wide association meta-analysis. And we did three different types of meta-analysis. So we did a non-stratified meta-analysis where we included sex as a covariate. And because we know that there are significant differences in performance between males and females, we also did a sex-stratified meta-analysis where we used only males in one of the meta-analysis, and we used only females in the other meta-analysis. In addition to that, we also did SNP heritability using LDSC. We investigated genetic correlation for a whole range of psychiatric conditions, psychological traits, and brain volumes. We were also quite interested in looking at sex differences because this was something that we observed at a phenotypic level. And finally, we also did functional enrichment and gene-based analysis. A small proportion of the people who completed the short version of the ICE test were twins. So we had 749 twin individuals, which was great because we could also use their data to calculate twin heritability. So I'll talk about some of the results over here. So when we look at the phenotypic scores, what we quite clearly identified was that there was a small but highly significant female advantage. So I think you can see that over here. Females tended to score slightly higher than men. But overall, the scores were similar to what we had previously recorded and reported in the general literature. We also knew that sex and age were significantly associated with scores on the ICE test. And in general, for both the short version of the ICE test and the complete version of the ICE test, the scores were normally distributed. Again, in both the short version and the full version of the ICE test, there was a consistent female advantage, so replicating some of the previous results. So we went ahead and performed GWAMA, so genome-wide association meta-analysis. As mentioned earlier, we conducted 3 different GWAMA, and we identified one SNP that was significant in the females only meta-analysis. And this was significant even if we take into account the fact that we are doing 3 different tests. There was one SNP, RS7641347, which had a GWAMA p-value of 4.8 to 10 power minus 9. But when we correct for Wiener's curse and we calculated total variance explained, this explained a very, very small proportion of the variance. So 0.013% of the variance, which perhaps is not very surprising because when you look at the literature and when you look at other complex traits, you see sort of similar SNPs explaining similar small proportion of variance. If we were to zoom in onto the SNP, we see that the SNP lies very close to a gene called LRRN1, which is leucine-rich receptor nuclease 1, neuronal 1. And when we query the GTEx data, we see that LRR1 has got very high expression. The highest expression of LRRN1 is in the dorsal striatum. And this becomes interesting when we look at some of the genetic correlations with brain volumes. We also looked at heritability estimates. So the plot that you see over here is the SNP heritability estimate or the narrow sense heritability estimate. So approximately slightly less than 6% of the total variance could be explained by all SNPs. And there was no significant difference between male SNP heritability or female SNP heritability, which was interesting. When we calculate twin heritability, approximately 30% of the total variance could be explained. And this was using an AE model. And if you were to stratify twins based on males and females and then calculate heritability again, we didn't identify a significant difference in twin heritability between males and females. So the real interesting results, at least for me, are in genetic correlations. We ran, as I mentioned earlier, we ran genetic correlations using summary GWAS data for 3 different types of conditions and traits. The first one that we ran were with brain volumes. And what you see is that there's a significant positive correlation between volume of the caudate nucleus and the ICE test. And similarly, a normally significant positive correlation between volume of the putamen and the ICE test. And interestingly, both the caudate nucleus and the putamen are part of the dorsal striatum. And as I mentioned earlier, the top gene that we identified, LRRN1, is one that has got the highest expression in the dorsal striatum. When we went back and looked through the literature, we saw that there were quite a few different studies, either leasing studies or brain imaging studies, which showed that the dorsal striatum was important in things like emotion recognition and cognitive empathy. There was one particularly interesting paper which reported a lesion in an individual in the dorsal striatum who was not able to identify emotions at all. And this was measured using the reading the mind and the ICE test. That we thought this was a very interesting validation of what we see in genetics. We were also very interested in looking at psychiatric conditions, and we identified a significant result for the anorexia nervosa GWAS. The correlation between anorexia nervosa and the ICE test was positive, which was very interesting because we hadn't expected that. In fact, if you look at all the different psychiatric conditions out there, so we have autism, bipolar disorder, depression, and schizophrenia, all the correlations are positive, even if only anorexia is significant. Now, when we went back and looked through literature, there's quite a mixed bag out there. There are few studies which show that individuals with anorexia or bipolar disorder, for example, tend to perform poorly on the ICE test. Certainly, quite a lot of literature on autism suggests that individuals with autism tend to perform poorly in this condition on the ICE test. But over here, in terms of genetic correlations, we are seeing a positive genetic correlation. And this was quite puzzling. And I think the way we think about this is because of 2 things. One is that the total variance explained by the ICE test, by all the SNPs in the ICE test, is only 5%. So the genetic information that we are capturing over here captures a very, very small proportion of the variance in the ICE test. So there could be other mechanisms that are not captured by the genetic GWAS data that we have over here, which can contribute to poorer performance on the ICE test in individuals with autism or depression. The other thing is the underlying pleiotropy. Certainly for autism, we know for certain that autism has got a strong positive genetic correlation with educational attainment and other measures of general cognition. As I will show in the next slide, we know that ICE test is also positively correlated with measures of cognition. So it's not immediately clear what will happen if we were to control for measures of cognition and then run the genetic correlation again. We haven't been able to do that, but it's something that we are thinking of running. And the final piece of evidence is that these conditions are incredibly heterogeneous at a phenotypic and a genetic level. There's one paper from our lab which shows that if you were to do hierarchical clustering based on the ICE test scores on individuals with autism, you see that individuals with autism cluster into 5 discrete groups. There are some individuals who perform better than typical controls, and there are some groups that perform worse than typical controls. We were also interested in a whole range of different psychological traits. Again, the most interesting and significant ones over here were with different measures of cognition. So we have childhood cognition, which was significant. We have college years, which was positive and significant. We have years of schooling, which was again positive and significant. And with the NEO5 factor, we see there's a significant positive correlation between openness to experience and the reading the mind in the eyes test. And when we go back and look at literature, when you look at epidemiological studies and psychometric studies, we show that there is a significant phenotypic correlation between different measures of cognition and performance on the eyes test. So it's nice that at a genetic level, we were able to mirror the correlations that you observe at a phenotypic level. The other bit of study that we were curious about was sex differences. So as mentioned earlier, there was a small but significant difference in between males and females in the eyes test. So there was a male disadvantage, or females tended to score slightly higher than males. So we wanted to investigate this at a genetic level. The first thing that we ran was a genetic correlation between sexes. So because we had sex stratified GWAS data, we were able to sort of use both the summary data sets and run genetic correlation using LDSC. And we see an reasonably modest, but not extremely high positive genetic correlation. So the genetic correlation that we see is only around 70%, but it's highly significant. And when I went to the literature and ran genetic correlation for other GWAS data where there was sex stratified summary GWAS data, most of the genetic correlations that I identified were close to 1. So we have on the measure on empathy, where the genetic correlation was about 85% between males and females. When you look at years of schooling, the genetic correlation is close to 92%. So in comparison with those, this is not as high as what you would expect. We also looked at it from a different angle. So we used all the LD Prüm SNPs and looked at concordance in effect direction between males and females. And 61% of the SNPs had concordant effect directions, which is similar to what you see at a genetic correlation level. But if we were to look at genes, so once you collapse the SNP-based p-values that we got from GWAS into gene-based p-values, and we look at the overlap in the normally significant genes between males and females, we do not find a significant overlap at a gene-based level. So again, we were not really able to tease apart what is happening at a genetic level in terms of sex differences. But I think there is some difference to show that at least part of the genetic architecture is distinct between sexes. We are thinking of this actively in slightly different ways. We are also looking at correlation networks, gene correlation, co-expression networks, and seeing if there's an enrichment over there. So in conclusion, we see a consistent female advantage on the eyes test. There's a modest heritability of approximately 30% when you look at twin samples. And when you restrict that to all the SNPs, you see that it explains 5% of the total variance. We identified one significant locus in the females only GWAMA, which was 3p26.2. There were significant positive genetic correlations with anorexia nervosa, openness to experience, and different measures of cognitive ability. And as I mentioned earlier, there was some evidence of sex-specific architecture, but we really need to dig into this a lot more to sort of understand what's happening. So this was an intensely collaborative project. So at Cambridge, thanks to Simon Baron Cohen, who's my supervisor, Bhishmadev Chakrabarti, Jyoti, Paula, Florina, 23andMe were phenomenal. They were able to provide us with all this data, and they were really amazing when we went back to them and asked them to run some of these additional analysis. So in particular, thanks to David Hines and Nadia Litterman. Thomas Bougeron is a senior author on this paper, and Roberto Toro, they were amazing. And thanks also to Dorrit Boomsma and folks at Amsterdam, and also Nick Martin, Sarah Medland, and Katrina Crosby, who contributed the PLTS cohort and did quite a bit of the analysis on that cohort. So that's it.
Thank you, Varun. This was very interesting.